Welcome to the deep dive. Today we're looking at a shortcut, really, to mastering something super complex in modern science. Computational biology. We're talking genomics, proteomics,
Right.
and the huge challenge of actually doing the data analysis.
Right. These fields, they depend on this massive ecosystem of specialized tools. Think of them like digital microscopes or centrifuges. They're absolutely essential for answering really deep questions about life itself. But working with them, it can be truly miserable.
And that's the core problem we're diving into. These specialized tools.
Mhm.
Maybe you're running a whole genome sequencing pipeline, right? Or
Yeah.
maybe something like chip-seq analysis looking at protein-DNA interactions.
Yeah.
They were all built for humans to type in these really specific, often unique command line instructions. Their inputs, their outputs, the way they technically connect or don't connect, it's all just fragmented, incompatible.
Exactly. It creates this, well, we call it the fragmentation barrier. And for an advanced AI agent, the kind of cutting edge system you'd want to run your complex research, it's almost impossible for it to talk to these tools
Yeah.
because they don't speak a common language. It's like the AI is fluent, but all the tools are speaking these ancient, completely different dialects.
Okay, so the mission for this deep dive, let's unpack the solutions, called the Model Context Protocol, or MCP. And specifically, we're looking at BioinfoMCP. It's this platform that, kind of amazingly, uses large language models, LLMs, to automatically convert hundreds of these difficult tools into standardized AI-friendly servers.
Yeah, and it's more than just like simple translation. This is about setting up a proper standard language for how tools and AI agents communicate. It basically gets rid of that steep technical learning curve. It means really complex computational analysis suddenly becomes accessible just using, well, natural human language.
All right, let's dig into that fragmentation barrier first. Just how bad is this fragmentation in the real world? Say a researcher is doing an RNA sequencing experiment, RNA-seq, trying to find genes that are expressed differently between samples. It's not just one program they run, is it?
Oh, definitely not. It's a whole pipeline, often end to end. You have to strategically link together multiple unique standalone tools. So you might start with something like FastQC for checking the initial quality of your sequencing reads. Then maybe use, say, Bowtie2 for aligning those reads to a reference genome, and then perhaps SAM tools to sort and index the results from the alignment.
And each one of those tools, built independently, have totally different commands, different ways of handling files. This sounds like the core of what you call the AI agent gap. General AIs, like the big LLMs we hear about, they're just too general for this, right? They can't handle the specifics.
Exactly. They're too broad. They get lost in the details, like the dozens of very specific subcommands you might need for a complex tool like JKK, for example. And even the specialized AI agents that do exist, people have built things like Bioplanner or AutoBA. They hit the same wall. They're smart enough to plan the analysis, figure out the steps, but they don't have a standardized way to actually run the tools they need. It's like having, I don't know, a brilliant chef who designs this amazing gourmet meal, but then finds out none of the kitchen appliances have standard plugs. The blender, the oven, the food processor, nothing fits the sockets.
We need that universal socket then. So that's where the Model Context Protocol, the MCP, comes in. What exactly is this protocol defining?
So MCP is basically that standardized language. It's for communication between an AI agent and a tool. When a tool gets processed by this BioinfoMCP system, it's converted into a standardized executable server. We call that an MCP server. And this server, it exposes all the functions of the original tool, but through a consistent, predictable interface.
Okay, so you've got the tool wrapped up as an NCP server. What does that plug into?
It plugs into any AI agent interface that also understands the MCP protocol. We call that the MCP host. That could be a platform like, say, Claude Desktop or maybe Cursor. And suddenly, the tool's functions are available through this simple, consistent API. Doesn't matter if the original tool needed a 5-line monster command or had an 80-page manual, it just works.
Wow, the value there seems huge.
Yeah.
So I don't need to remember the specific kind of arcane command for SAM tools to sort my alignment file anymore. I can just tell my AI host, hey, sort these aligned reads, and the MCP server figures out the rest.
That's the idea, exactly. It handles the translation for you, and that really democratizes access. Suddenly, a biologist who maybe hasn't ever used the command line can actually run these highly sophisticated analyses. But here's the kicker, as you might guess. You have hundreds of these tools in bioinformatics, maybe thousands, and they're constantly being updated. Manually converting all of them into MCP servers, that's just impractical. It's unsustainable. It would take teams of developers years. And that critical bottleneck, that scaling problem, is precisely what BioinfoMCP aims to solve with automation.
Okay, so let's get into that automation, the BioinfoMCP Converter. How does it actually automate this conversion using the LLM engine? What are the steps?
Well, it starts with preparation. The system first needs to get its hands on the tool's documentation, all of it. Usually that means either feeding it a downloaded PDF manual if one exists, or actually running the tool's own help commands, like typing help or AH on the command line to get the usage instructions. And a key thing they found here is that the quality of that original documentation, how clear and detailed it is, directly impacts how well the automated conversion works. Garbage in, garbage out, basically.
Makes sense. The LLM needs good source material to work from.
Yeah.
So once it has ingested all that documentation, what happens in the next stage? Execution.
Right. This is where the LLM really does the heavy lifting. It uses a pretty robust LLM backbone. The LLM generates the Python code needed for the MCT server, and it does this using a specific internal framework to keep things consistent. But then, and this is clever, the system doesn't just trust the generated code. It becomes its own quality control check, its own harsh critic, you could say. It rigorously checks for two main failure points. Did the LLM actually generate any code? And does the code it generated have any syntax errors?
Ah, so it's kind of forced to debug itself, like a compiler that just keeps trying until the code is actually valid?
Pretty much, yeah. If it finds errors, it automatically triggers a loop. It tells the LLM, okay, that didn't work, try again, fix these issues, and it refines the code. It iterates until the syntax is clean. And then finally, you get to the delivery stage. The refined, working MCP server code gets neatly packaged up into an executable Docker container, which is just a clean, self-contained little server ready to be deployed and connect to any MCP host.
That sounds pretty robust, but making an LLM reliably generate working code from, often dense technical manuals, that must require a very carefully designed prompt, right? How did they structure that prompt to get predictable results?
Oh, absolutely. The prompt engineering is critical here. It's very structured, built around 4 key parts: role, task, instructions, and requirements. The role basically tells the LLM, okay, you are now a specialized bioinformatics software engineer. Sets the context. The task gives the general goal, like, your job is to extract all the command line parameters from this documentation, figure out their types, are they required arguments, optional flags, do they need specific file types, things like that.
Okay, and the instructions part must be the real step-by-step guide, telling it how to write the code.
Exactly. It gives very specific directions. How to handle file inputs, how to actually execute the original command line tool using what are called sub-processes internally. And critically, it insists that the LLM must capture both the standard output, the stout, and the standard error, the starter, from the tool's execution. We'll probably come back to why that's important. And then the final part, requirements, is like the strict rulebook. It enforces standardization. For example, it demands that all parameters are handled with the correct data types, and it might prohibit certain general programming tricks that can make the server less reliable.
I saw in the research notes that picking the right LLM model was also a big deal. They didn't just go for the fastest or the cheapest one. What was the key technical feature they really needed?
Yeah, the absolute crucial factor turned out to be the context window size. You see, a lot of these bioinformatics tools, especially the really powerful ones like the ISPADES genome assembler, or even just certain sub-commands within big toolkits like SAM tools, they have massive documentation, really long, detailed manuals. So they found they needed an LLM with a huge context window, something that could handle up to 1 million tokens of input text. If they used smaller models, even if they were faster or cheaper, the model would essentially forget or ignore parts of the documentation halfway through. That led to buggy or incomplete MCP servers.
Ah, okay. So it's like needing a model that can read and remember the entire textbook, not just skim the summary on the back cover.
Precisely. And focusing on that documentation capacity, that large context window, allowed the BioInfoMCP Converter to work really well. They successfully converted 38 pretty diverse bioinformatics tools, including some complex ones with multiple functions like GATK or deep tools. And the average conversion time was remarkable, around 40 seconds per tool, with very little human help needed.
Okay, so you've automatically built these MCP servers. But how do you know they actually work reliably? Building is one thing, validation is another.
Right. That's where the BioinfoMCP Benchmark comes in. It was designed specifically to test that these automatically converted tools are robust and versatile enough for real science.
How do they test that? You can't just run one command and say, yep, looks good.
Right.
Especially if the LLM wrote the code.
No, definitely not. They used a two-part strategy for validation. Part one is individual tool testing. This is like a basic sanity check. Does each individual MCP server run its command without crashing internally? And crucially, do the output files it produces exactly match what you'd get if you ran the original tool by hand, the old-fashioned way? This ensures the basic building blocks are solid.
Okay, that makes sense. And part two sounds like the more complex test, the pipeline execution testing.
That's right. This is the real-world stress test. Here, they give AI agents complex tasks that require chaining multiple MCP servers together. Like that whole RNA-seq analysis pipeline we talked about earlier, quality control, then alignment, then sorting, maybe even more quality assessment downstream. The AI agent has to manage that whole sequence.
Now, for the AI agent to manage that chain, especially if something goes wrong, it needs feedback from each tool, right?
Yeah.
You mentioned that structured JSON output earlier. What information is in that?
Yes, that JSON output is key. When an MCP server runs a command and finishes, it sends back this neat little package to the AI host. It contains 3 essential bits of information. First, the exact command executed, so you know precisely what was run. Second, the stutter, which is the standard error stream. And third, the stutter, the standard output stream.
Could you quickly explain for listeners maybe not deep into command lines, what are stutout and strutter?
Sure, think of them as 2 different channels for text output from a program. Stateout is usually where the main results go, the data you wanted, the summary report, the confirmation that everything worked. Stutter is typically used for messages about the program's execution status updates, warnings, and importantly, any error messages if something went wrong.
Got it. So why is getting both of those packaged up nicely in that JSON so important for the AI agent managing the pipeline?
Because it lets the AI do more than just blindly run the next command. It allows the AI to understand what happened. Take that FastQC quality control tool again. The AI gets the JSON, reads the output streams. It can see the warnings or critical issues reported by FastQC, and then it can immediately summarize that for the user, maybe classifying things into critical issues found or warning issues to check. It provides actual insight right away based on the tool's own feedback.
Okay, let's walk through that complex RNA-seq pipeline example again. If something breaks midway, say the alignment step fails for some reason, how does the AI use that JSON feedback to adapt?
Great question. Because the AI host gets the full JSON, including any error message that the tool wrote to Strutter, it knows why it failed. Maybe an input file was missing, or maybe the data wasn't sorted correctly for the next step, like running QualiMap for quality assessment. The agent sees the error, understands it, and can then modify its plan. It might realize, I need to run SAMTools sort first, execute that step, then reattempt the QualiMap step that failed before.
That adaptability, that's what makes it truly agentic, isn't it? It's not just a fixed script that breaks on the first error.
Exactly. It can react and potentially recover. And the benchmark results really back this up. They found that 94.7% of those 38 converted tools could be successfully used in these complex workflows by different AI agent platforms. They tested a local one, Claude Desktop, and Cursor. It showed the standardization really works across the board.
This really does feel transformative. So for the actual scientist, the person doing the research,
Yeah.
what's the immediate impact, that steep learning curve we mentioned? Is it just gone?
For many tasks, yes, it's effectively demolished. Researchers can shift their focus. Instead of spending hours or days trying to memorize or look up obscure command line syntax, they can interact using natural language. They just describe the scientific goal they have. And the BioinfoMCP system, through the AI host and the MCP servers, translates that goal into the necessary sequence of technical steps. It massively boosts productivity simply by removing that friction, that barrier between the scientific idea and the computational execution.
And it seems like there's more than just a speed benefit. You mentioned the AI interpreting results.
Yeah, that's a really interesting aspect. Because you have the LLM's reasoning capability in the AI host combined with the standardized, detailed output from the MCP servers, scientists can get an immediate, structured interpretation of the results. Before they even dive into the full raw output files, the AI can give them a summary, point out potential issues based on the stutter warnings, or highlight key findings from the stutout. They get actionable insights much faster.
Okay, it sounds incredibly promising. But we should probably also talk about the current limitations. What are the hurdles that still exist?
Right. It's important to be realistic. There are definitely constraints. First, there's the documentation retrieval part we touched on earlier. Right now, the system still needs a bit of human help to get the latest documentation for a tool. You might have to point it to the right PDF file or manually run the help command. The future goal is clearly to make that fully autonomous. You just tell the AI the name of the tool, and it goes and finds the relevant manual itself. But that's not quite there yet.
Okay, that sounds like a software challenge, probably solvable over time. Yeah. What about the actual computing power needed? Bioinformatics deals with enormous data sets.
That's the bigger, more fundamental bottleneck right now related to hardware. These AI agents, running through platforms like Claude Desktop or others, they tend to struggle with tools that are extremely memory intensive or take a very long time to run. Think about really demanding sequence aligners like STAR, which are workhorses in genomics. The core reason is that these current AI agent systems generally can't connect to or utilize graphics processing units, GPUs, and GPUs are absolutely essential for accelerating these massive computational tasks.
So even if BioinfoMCP perfectly converts a tool like STAR into an MCP server, running a huge alignment job through the current AI agent interfaces might just time out or run out of memory because it can't access the necessary GPU power.
Exactly. For those really heavy-duty jobs, you likely still need a human to set up and run the execution in a dedicated high-performance computing environment, probably with GPUs. The software side, the interface problem, has been largely solved by MCP. But the hardware performance for the most demanding tasks remains a constraint for purely AI-driven execution for now.
Understood. But even with that specific limitation for massive jobs, what BioInfoMCP has accomplished seems huge. It really has bridged that enormous gap between all these incredibly specialized, historically fragmented bioinformatics tools and the modern world of AI agents. It's turned this landscape of incompatible scientific silos into something more like a unified, accessible library that you can interact with using human language.
Absolutely. It signals a potential revolution, really, where deep technical command line expertise might no longer be an absolute prerequisite for doing profound computational science.
Which leads us nicely to our final thought for you, our listeners. If this kind of LLM-driven automation, using a standard like the Model Context Protocol, can bring order to the famously fragmented world of genomics and proteomics, what other complex, specialized scientific or technical domains might be out there? Think about chemistry simulation software or materials science modeling tools, maybe even complex financial trading systems or niche manufacturing control software. Fields that are currently locked behind incompatible interfaces and steep learning curves. Are they also ripe for an MCP-style revolution? What technical field do you think is desperately waiting for its own AI-powered conversion tool next? Something to ponder.